Good morning, students. Today we will start the chapter Mitochondria. So, I am sharing the screen. As we know, mitochondria is called as the powerhouse of the cell because it forms ATP, a high energy rich currency inside the matrix, and it is a filamentous, granular, light microscopic, as well as self-replicating organelles. Self-replicating because it has its own DNA and the site of cellular respiration, as well as semi-autonomous in its nature, and forms the ATP, energy currency, inside its matrix. So it is also called as the powerhouse of the cell. Occurrence and distribution. Basically, mitochondria, these are absent in prokaryotic cell like bacteria, cyanobacteria, PPLO, and present in all eukaryotic cell except mature and old mammalian RBCs. In most of the cell, it is distributed uniformly in the cytoplasma, and basically it is present in abundance in metabolically active cells and muscle cells, where they perform the specific functions like human sperm. In the axoneme part, we can see a spiral chain of mitochondria present in it because it have to perform the high motion locomotion inside the fluid, and its abundance are also seen at the synaptic cleft region because the synaptic transmission of nerve impulse takes place here. And the shape and size of mitochondria, it varies. Mitochondria is pleomorphic in nature. Pleomorphic means its shape is variable. As we can see, it is granular and filamentous in many organisms, while trypanosomas, they show loop-like mitochondria having DNA-rich fibrillar bodies called as nitoplast. So its size shows great diversity. We can say it is the second largest cellular structure present in a eukaryotic cell. The smallest sized mitochondria are present in a yeast cell, while the largest sized mitochondria are seen in mammalian pancreatic cell. This is the largest cell organelle in animal cells, while second largest organelle in plant cells. It is the third largest structure present in plant cell after nucleus and chloroplast, then mitochondria. The number of mitochondria varies not only in different organisms but in different cells of the same organisms. It depends on whether the cell is metabolically more active or not. These are more in growing, dividing, and metabolically active cell, so these are present in larger amount there. All the mitochondria present in a cell are collectively called as chondriosomes. The maximum number of mitochondria are seen in flight muscles of insects, which are collectively called as sarcosomes. In case of plant cell, less ATP is formed inside the mitochondria because chloroplast form the ATP. Now we will discuss the ultrastructure of mitochondria. This is the ultrastructure of mitochondria. It shows two limiting membranes and two chambers. Firstly, the mitochondrial membrane. These are two in number, the outermost and innermost. Mitochondria basically is a bean-shaped structure. The outer membrane is thick, lipoproteinous, and trilaminar unit membrane structure. It have increased lipid molecules and less proteinous content. It is freely permeable because of the presence of transmembrane channels called as porine proteins, which act as sieving action and allows the diffusion of 500 Daltons molecular weight freely from cytoplasma to the mitochondria outer chamber. It also contains certain enzymes which performs the elongation of fatty acids, oxidation of epinephrine, and degradation of tryptophan. Unlike the inner mitochondrial membrane, which is showing slightly zigzag pattern, this outer membrane is smooth and asymmetric in nature. It is in contact with the cytosol at certain points and can be continuous with membrane of endoplasmic reticulum. These are mitochondria which having no outer membrane are called as the mitoplast. Here we can see, this is the diagram. In it, the outer membrane, it shows pore like structures present on it. This is the outer membrane, and the circular part I have mentioned it. It is the intracrystal space. What is crista? Crista are the compartments present in the inner membrane. These are formed by the involutions of inner membrane. We can see there are ribosomes of 70s. We can see here that ribosomes present in mitochondria are equivalent to prokaryotic cell. Here we can see a bulb-like structure that is called as oxysome present on inner membrane. The dense granules are present here, which are heterogeneous in nature, containing various enzymes, various cofactors, and divalent ions. The space between the outer chamber and inner chamber is called as intermembraneous space or outer chamber, while the space present inside the mitochondria is called as mitochondrial crystal space, intercrystal space. The matrix contains various enzymes responsible for protein synthesis, lipid synthesis, and Krebs cycle. We will discuss all this in later topic. Now, here we can see that this is the diagram of inner mitochondrial membrane. Here I have shown this diagram in an enlarged way. This bulb-like structure is called as basically oxysome, having headpiece, stalk, and basal piece. This outer membrane and inner membrane, these are separated by each other by an outer chamber. We can see that these oxysomes, these are embedded inside the inner membrane, and the inner membrane have involutions called as intracrystal space. The bulb-like structure are embedded in it. The basal part of oxysomes is called as base piece or F0 particle. It is also called as cytosolic phase, while the bulb-like portion, it is floated in matrix or also called as M phase. Now, the inner mitochondrial membrane, it is thick, lipoproteinous, and have more protein content as compared to the outer membrane, which have lipid content higher. It also contains various carriers and permeases as semi-permeable membrane, and a number of folds are present which are called as mitochondrial crest and crista. These are formed by the involutions of inner membrane. These folds can be unbranched or branched, complete or incomplete, tubular shape and villi shaped. These are basically of two types: septate crista, which are parallel sided and usually have single and straight fold, while tubular villi, tubular crista, which appears as villi like a partition. These terminals ends may have swollen ended. Here we can see that these crista increases the surface area for the enzyme activity which are present in cellular respiration. The higher surface area provides large energy production, and we can see that the crista are studded with a variety of enzymes like cytochromes, enzymatic proteins, and ATP synthase. The mitochondrial crista are homologous to the mesosomes of the prokaryotic bacteria, as I have shown in the figure side. This is the prokaryotic cell, and the inner membrane of bacteria contains small granule-like particles around its plasma membrane, which performs the function of ATP synthesis. They have no well-organized organelles like mitochondria and nucleus. They have only scattered granules which perform this function, and here the function of ATP production is performed by mesosomes. So we can say that eukaryotic mitochondria is equivalent to prokaryotic mesosomes. The number of mitochondria and their size, it depends upon the metabolic rate of the cell and its growing time period. For example, germ cells, liver cells, muscle cells and neuronal cells, these have to perform much higher work, so they have abundance of mitochondria. Now we can see inside the mitochondria, each crystal are separated by a space. This is called as intercrystal space, which is continuous with outer chamber. And this crystal divides the mitochondria into a number of interconnected chambers. Means all these chambers are connected by this outer chamber that is present between outer membrane and inner mitochondrial membrane. The arrangement of crista can be differs, but most common crista arrangement is perpendicular to the longitudinal action of mitochondria. This figure 4, it shows various types of crista or involutions which are formed during inner mitochondrial membrane. Here we can see. Now, the inner mitochondrial membrane is studded with oxysomes. It is also called as F1 particle or elementary particle. The base is denoted by base piece, or we can say it as F0 subunit. It is rectangular in shape and embedded in mitochondrial membrane, and this baseline, it contains a proton channel. Now the second part is called as neck or stalk. This is oligomycin sensitive conferring protein OSCP, and it also contains F5, F6 and FC2, which binds to inner mitochondrial membrane. And the headpiece, it is also called as F1 subunit. It is spherical in shape and it contains ATPase enzyme or ATP synthetase. It controls the synthesis of ATP by phosphorylation of ADP. So this oxysome is also called as ATP particle, and these are the functional unit of mitochondria. This F1 particle or elementary particle, these are also called as Fernandez-Morin subunits, because these are discovered by this scientist, and these are evenly spaced at the interval of 100 angstrom. Each oxysomes have these following parts. Now, it is suggested that enzymes for electron transport chain and oxidative phosphorylation are located in these F0 and F1 particles. I have shown in the diagram that complex 1 and 2, these are present in the base piece, while complex 3 is present in the stalk, and complex 4, that is present in the F1 particle in the head of this bulb. Now, there is a difference between the outer and inner mitochondrial membrane. Like the outer membrane, the crista are absent, it is smoother, while in inner membrane the crista are present and it is zigzag shaped. The outer membrane shows freely permeable, while inner membrane is semi-permeable. The protein content, it is lesser in outer membrane, while 80% of inner membrane is consist of proteins in it. Many of these are associated with electron transport system and act as carriers. The lipid content present in the outer membrane are very less, and the cholesterol, it is also in lesser abundance, while in inner membrane the cholesterol is not present there and large amount of cardiolipids are found. The presence of salic acid is seen in outer membrane while it is absent in inner membrane. So mitochondria chambers are two in number. Number 1 outer chamber and number 2 inner chamber. Outer chamber is intermembranous and perimitochondrial. It lies between the two chambers, and it is filled with less denser granular particle which is heterogeneous in nature, having water, minerals and enzymes. And one important thing that its medium is acidic in nature. Why it is acidic in nature? Because of the proton gradient which is produced due to the pumping of protons from the mitochondrial matrix into this space via electron transport chain. Now, inner chamber, it lies inside the inner mitochondrial membrane and filled with more dense gel-like and homogeneous mitochondrial matrix. It is generally granular and having many divalent ions binding sites like magnesium, calcium, and other mitochondrial matrix having many cofactors in it. Now, the Mito Ribosomes, these are the Mito Ribosomes are basically 70S in nature, or we can say these are 55S ribosomes present in the mitochondria. The mitochondrial DNA is small, circular, double-stranded and naked like prokaryotic DNA. It is linearly organized in cnidarians. The mitochondrial DNA is fusion positive, means showing a positive staining with fusion staining, and consists of 1% of total cellular DNA. It have genetic information and rich content of GC pairs, and encodes more genetic information in the form of proteins. Near about 37 genes are encoded by mitochondria. It is semi-autonomous, and the DNA containing areas of mitochondria are called as nucleoid. Mitochondrial genome shows a remarkable similarity with the bacterial genome.